Okay, I guess why don't we get started? Any questions? Everybody's doing okay? Hopefully we're gonna finish off pH today, and we're gonna talk about buffering, and then we're gonna talk about some control, some sort of liming controls and stuff like that. But I wanna finish off last lecture because I didn't get all the way through buffering, and I wanted to, because it's a fairly important process, and certainly from a soil management perspective, it's extremely important process. But in general, just from issues of nutrients and things like that, buffering is a really, it's a driver for a lot of the reactions that are going on in soils. Certain pHs, or I should say nutrients, have certain availability dependent on different pHs, and if you have a buffering capacity or the ability of soils to resist pH change, that's going to have an effect upon the availability of the nutrients. And so I started talking about buffering with this slide because when we're talking about buffering, we're talking about controlling or managing, perhaps that's not the right word, but controlling all of the pHs. We're not just controlling this active pH. Yes, this is the pH that we measure, and this is the one that controls the availability of nutrients in a sense, but because all of these different pools of pH are in equilibrium, when you want to control the pH of your soils, this is what you're interested in, but you have to also be aware that because of this exchange, you really are controlling these two pools as well. Okay? Now, I started with looking at this because this is actually something we looked at last lecture when I was on this board over here. But the size of that reserve, or in essence, another way of saying that the size of the cation exchange capacity is going to have a huge impact on the salt replaceable or the exchangeable pH pool. Does that make sense? Okay. The larger the CEC and the more bases or the less bases, the larger the base saturation, the more exchange I'm going to have between these two pools. Okay. If this is a very small CEC, cation exchange capacity, and it has a very low base saturation, most of the cation exchange sites are going to be occupied by aluminums or protons, which even if I have exchange, it's going to be a proton for a proton, right? On the other hand, if I have a high base saturation, I start having calciums and sodiums and things like that, magnesium in these cation exchange sites, when the proton exchanges, I'm going to get something other than a proton coming out. And so that's going to affect the pH in this active pool. Does that make sense? Question?
Why, when there's a low CEC and low base saturation, is the aluminum on that surface?
Why? Say that. So why, when there's a low base saturation
And low CEC,
on
the surface of the...
Oh, right. It's not necessarily there's just because I have low CEC or high CEC doesn't necessarily mean that there's going to be aluminums on these exchange sites. But once I start getting down to low pHs, all the other bases are gone, and what's really left is aluminum and protons. Okay? Aluminum will be rinsed out of the system as well, but because aluminum doesn't react to reduce the acidity as the other bases do, because aluminum, you put alumina into a solution and it actually increases acidity. Until you get down to really low pHs. Which brought us to this issue of a slide like this, where I had three different treatments. Okay, one treatment was a treatment with a high base saturation, high CEC. Another treatment was a soil that had a low CEC, and the third treatment was water, where there's no CEC whatsoever. Okay, and imagine that the low CEC and the high CEC, both of those soils have high base saturation. Let's imagine they have 100% base saturation. It doesn't really matter. Okay? But what does matter is the amount of protons that you're adding. Okay? If I have a soil that has a high base saturation and has a high CEC, it has a huge amount of resistance to pH change, because as those protons start taking away the bases that are on the cation exchange, exchanging for them, I'm going to have a very large pool of bases here. Now, if I have a smaller pool, i.e. a low CEC, even with 100% base saturation, I'm going to have less bases that can exchange. So my resistance to pH change is going to be greatly reduced. And then in the third scenario, this scenario right here, which is water, basically every proton I add is going to increase acidity. There's nothing to exchange. It can't exchange on any of those exchange sites. Every hydroxide I add is going to raise the pH. It's a one-to-one correspondence. Does that make sense to everybody? Now, there are controls on this change in pH at different pHs. Now that sounds strange. Okay? There are different controls on the buffering at different pHs. Okay? At the mid-level, between basically 4.5 to 7.5, because there are a lot of bases still on these cation exchange sites, the bases themselves act as a buffer to pH change. I start adding protons out here. Those protons are going to exchange with the bases. The bases are going to go into solution, and the active pH is not going to change all that much. Does that make sense? Now, I could also add bases over here. If I start adding bases, the assumption is, well, the pH is going to go up. No. The bases are going to start exchanging with the protons, and the proton's going to get back in to the active soil solution. And so they're resisting change. Yes, there will be a change in pH, but it won't be as dramatic as if I was doing it in water. Does that make sense to everybody? And that control is strictly associated with the cation exchange. It's with those bases, the base saturations on those cation exchange sites. Okay? Now, when we start getting at the extremes, the high pHs and the low pHs, there are two other controls. At the high pHs, this is where we ended up last lecture. At the high pHs, you're really looking at calcium carbonate. Okay, so do you guys remember this equation right here? CO2 plus water makes carbonic acid, right? Okay, now this is a reversible reaction. This reaction can go this way. Now, at high pHs, we're really at this component of the pH reaction. Okay, so at high pHs, we have a soil solution or a soil. Here's my soil, all right? And in this soil is calcium carbonate. Right? Now, if I start to acidify this system, I'm going to be adding protons to it, right? What's going to happen? This reaction right here. Here's my calcium carbonate, and here's my acid. So for every 2 protons that I add into my calcium carbonate enriched environment, those 2 protons are basically going to knock the calcium off. One of those protons is going to bind with the carbonate, and one of those protons are going to be in solution. So for every calcium carbonate I have, for every 2 protons that are added, it's going to neutralize one of them. It's not going to prevent acidification, but it's going to slow it down. Does that make sense? Questions? Go.
I have a question about the last slide. If I heard correctly, you said that you add bases to the solution and then they interchange with protons. But if you add base cations, you acidify the soil?
In a sense, this buffering action, the question is, he didn't basically understand when he started adding bases what's happening. Okay? So this reaction is totally reversible. If I add protons and I have a high base saturation, those protons are going to knock the bases off their exchange complexes, and it's going to remove a proton from the soil solution for every exchange, right? So as a result, the active pool, the active acidity, is resisting pH change. Okay? I keep driving it down, protons, add more and more protons, and sooner or later I'm going to get really low pHs. Now let's turn the scenario around. I've got the low pHs. Now I'm going to start throwing bases in. Well, those bases are going to go into the solution. What are they going to do? They're going to equilibrate with cation exchange sites. If they do that, and the cation exchange sites are dominated by protons, what's going to be coming off with the exchanges? predominantly it's going to be protons. And so every base that I throw in, it exchanges, in all likelihood, a proton is going to be coming out. So it's actually going to resist the raise in the pH because I keep throwing protons back into solution for every base that I'm throwing into solution. Does that make sense? Okay? All right. So there was another question here on this one. Was there not? We feel comfortable with this? All right. So let's actually start talking about aluminum. Now this is at the low pH. This happens at the high pHs. This happens at the low pHs. Okay? And this is this slide, which I know everybody just loved. Okay? Remember this slide? This is the slide that was talking about how we get protons in solution. Okay, let's start here again. Aluminum, it's a part of the structure. Okay, protons go into solution and they start dissolving this clay micelle. Aluminum goes into solution. Okay, and I've got a smaller clay micelle. Okay, if I am at this scenario down here with this high acidity, I'm putting aluminum into soil solution. The aluminum is probably not going to do that much. Okay, at low pH is high concentration of acidity, high concentrations of protons. Aluminum is basically sitting all by itself in +3. Okay, so we have a low pH. In fact, the pH probably lower than 5. Now, what's going to happen when I start trying to raise or change the pH? What's going to happen when I start adding hydroxide? This aluminum is going to bind with that hydroxide to make aluminum OH plus 2. Okay, so it's literally going to take that aluminum out of the soil solution. All right, let's go back over here and take a look at this. Same soil, but in this soil, I have an enrichment of aluminum. Okay? And into that soil solution, we're going to be adding hydroxide. I want to raise the pH. Now, we will talk about where that comes from in a moment. But in essence, to raise the pH, I need to add hydroxide. Well, what is this going to do? This is going to react with that. Okay? And I'm going to get OH minus. This OH is going to combine, and what's going to happen is I'm going to get this. In essence, I've taken that OH out of the soil solution, which means I'm resisting pH change. This aluminum, by combining with this, is taking it out of the soil solution, and therefore it's resisting the pH change. Does that make sense? Now, okay, so I'm going to add 2 of these. I'm going to add another one. I add another hydroxide, OH. This combines with this. And in essence, I've neutralized another hydroxide in the soil solution. I'm resisting the raise in the pH. I do another one. It will combine with the aluminum over here, make aluminum hydroxide. In essence, this one aluminum neutralized 1, 2, 3 hydroxides. It's buffering the pH. It's resisting the rise in the pH. Go.
So this diagram suggests that it's taking the hydroxides from water molecules?
This diagram is designed to show both directions. Okay, so we get up to here. This is the Gibbsite form. Okay, if I've been adding hydroxide again and again and again, okay, what you really should think of is this, the proton, what's being released out is the hydroxide. This one, the proton's being released. Okay? But what you're looking at here is we've driven from here with a hydroxide to here, with a hydroxide to here, with a hydroxide to here.
Right, but if the hydroxide is coming from the H2O, then how is it buffering? Isn't it not? It's not affecting.
In this scenario, this is the whole reaction. I'm just showing you part of the reaction. Okay? Now, we could do this the other way around, because this, in fact, will help. This, in fact, will help to buffer the reaction the other way as well. Okay? So what happens when I start throwing protons into the soil solution? Hydroxide gets stripped off, turns into water, and we now have that. So the proton, in essence, has been neutralized. So let's throw the scenario the other way. I start throwing protons in here. The byproduct of that proton, let's show the whole equation, is water, and I now have that. Completely reversible. I do it again with a proton. Oops. Proton makes water. I've neutralized another one. I can do it one more time. Proton, aluminum. So in essence, this, depending upon what pH you are in, this guy can reduce or neutralize 3 hydroxides as it moves up and 3 protons as it moves down in pH. And the pKa that you're seeing here is the approximate pH at which this happens. So this buffering reaction is happening someplace around 6.5 to about someplace around 4.7, 4.5. Okay? So the buffering reaction in this case is the lower ends. It's bracketing this zone. Does that make sense to everybody? Questions? Cool beans? We're good to go? All right. Skip over that one. Well, let's go back to this one. All right. So these are simple buffer reactions. They happen normally in soils. Okay? But from a management sense, we might in fact want to raise or lower the pH intentionally. To do that, we need to think about this whole issue of the reserve acidity. We need to figure out what actually, we need to know what the total acidity is. We need to know what the pH is of our active soil solution, the active pH, but we also need to know what's going on with the exchangeable as well as that reserve, the residual, okay, or the structural pH. Because I need to be able to deal with both the stuff that's in the active solution, but I also need to deal with the stuff that's going to be bouncing off the cation exchange sites and potentially what's going to be weathering out of the structure, right? So I really need to know what the size of these reserves are. If it's a well-buffered soil versus a poorly buffered soil, it's going to be a lot easier to change the pH of a weakly buffered soil than it will be a well-buffered soil. Does that make sense? Yeah? Okay. So we need to figure out what this buffering capacity is. We need to be able to deal with the soil's capacity to release or neutralize aluminum protons and the total amount of bases or acids that are needed to bring about that specific shift in pH. The best way to visualize this is something like this, where here's the pH that I want to move to. I want to get up to that pH. My pHs are now at this point. Okay? And basically, how much calcium carbonate, in this scenario, I'm trying to raise the pH, how much calcium carbonate I need to add per amount of soil. Okay? The poorly buffered soil, A, this poorly buffered soil versus the well-buffered soil, the visualization basically says, and tell me if I'm wrong, if this doesn't make sense, but I need to add less calcium carbonate to deal with soil A to bring it up to the pH the desired pH, in this case 6, versus soil B. Now, there's one other piece of information here. You have to understand what your liming agent is as well. It's not just about your soil, but you also want to have an understanding about the strength of your liming agent. Okay, there's lots of different liming agents out there. This is a list. This is from your textbook. I don't expect you guys to be able to see this right now, or be able to read it right now. But there are a lot of different types of liming agents out there with a lot of different strengths. Calcium carbonate is just one type of liming agent. You can have calcium hydroxide. It's twice as powerful as calcium carbonate. Okay? So a calcium carbonate will neutralize 1 proton per 1 calcium carbonate. Calcium hydroxide will neutralize 2. Okay? This is calcium carbonate. This is calcium hydroxide. This guy will neutralize 1 proton. This guy will neutralize 2. So for a 1-for-1 ratio, I'm getting in 2 times my bang for my buck here. Right? Make sense? Okay, how comfortable are we with this at this point, guys? Questions? Okay, well, let's think about one. I got one more slide here, and let's sort of think about this. We'll walk our way through this. Okay, at the high pHs, okay, certainly calcium carbonate will act as a buffering reagent. Okay, at low pHs, we basically throw it in there because we want to raise the pH. Okay, so this is the liming reaction. I'm missing a bracket here. Sorry, there should be a bracket right here. This is the carbonic acid. Okay, now, if we throw calcium carbonate into the soil, an acidic soil, what's going to happen? The proton is going to interact with this calcium. The calcium is going to go into solution. One of the protons is going to be joining onto the calcium carbonate, the carbonate component, and one of them is going to stay in solution. So what have I done? What have I done? What? I've raised the pH by neutralizing one of the protons. Now, if I want to raise it more, I'm going to have to add more calcium carbonate. And that's where this slide comes back into vogue. Basically, it tells you how much calcium carbonate I need to add per kilogram of soil to add it or to raise the pH. Now, you notice that there is no numbers here. How much is that? How much is that number right there? How much is that number right there? Well, that's going to be based on the buffering capacity of the soil that you're trying to raise. Okay? The scenario that we're showing here is 2 different soils. One's well buffered, one's poorly buffered. But there's going to be a whole spectrum of different types of soils out there, and you basically need to figure out how much I need to raise it to a certain amount. Now, there are basically 2 ways of doing that. There's a laboratory method, which you guys are going to be learning in the lab. And basically what this is a titration where you take a certain amount of soil and you add a certain amount of calcium carbonate or whatever your buffering reagent is, and you basically watch how far it raises with a certain amount of addition. Does that make sense? Okay, you calculate and you just fit the curve and you can say, okay, well, I have 2 acres and it took this much to raise this beaker full. How much do I need to raise it for my rooting depth? Okay? The other one is basically a rule of thumb, and it works fairly well. But the reality is this is not going to be perfect. The coarser the texture, this makes sense. The coarser the texture, the lower the CEC. So the lower the CEC, forget about the bases, the lower the CEC, one would assume the lesser amount of calcium carbonate I would need to add to raise the pH. Because we're basically assuming a certain amount of base saturation based on the textures. Now that is a poor assumption, but generally your soils will reach an equilibrium in the soil system out in the real world, and this sort of rule of thumb tends to work. Okay? Questions? You cool? All right. Take a look at this sheet. It's in your book. I don't expect you guys to be able to read it right now, but all right, good. Then we're going to stop here and we're going to start with so we've been asking where does the proton come from, or where does the acidity come from, and we've also been asking, what's the distribution? Okay? So the next thing I'd like to ask is what is going on in the environment to produce a specific pH at one place versus another, and what are the consequences of that pH? Okay, so we don't have to do the buffering reprise because we just basically did it. So what are those consequences? There are a number of different consequences out there. I'm going to be talking primarily about biological effects, but there are a number of different effects out there. From a biological sense, plant growth, nutrient availability, as well as some deleterious effects, primarily toxicity issues. But some of those other effects, certainly as the soils get more acidic, what am I going to start seeing weathering-wise? I'm going to see an increase in weathering. If I see an increase in weathering, I'm certainly going to see an increase in nutrients, but I'm also going to potentially be watching a lot of those nutrients leave the system. Okay? Certainly as the pHs go up, I'm certainly going to start seeing toxicity issues. Okay? Now, let's start with plant growth. And to start with plant growth, I'm going to start with this slide right here. Okay? The top part of this basically represents acidity. The blue line represents proton concentration. This black line basically represents hydroxide concentration. As the protons increase, the pH is going down. Okay? Now, interestingly enough, there's a direct correspondence to pHs and nutrient availability. Okay? This is overall nutrient availability. Now, how many different nutrients are there? Macronutrients and micronutrients. We're basically looking at all of these in total, and you'll notice as this line goes up, there's more and more availability. You'll notice that someplace around 6.5 is the maximum amount of total nutrient availability. That's not to say that one nutrient, it's not to say that every single nutrient is at its maximum availability. It's all of the nutrients in total combined. What's the maximum availability? And it happens to be around 6.5. Interestingly enough, this is not super surprising, as you get a plant response. Maximum plant response, someplace around 6.57, because that's where the maximum nutrient availability is. Kata. It depends on the plant. Certain plants have different nutrient needs, but overall, when you look at all the plants, combine all the different varieties of plants, agronomic plants as well as non-agronomic plants, trees versus herbaceous, annuals, perennials, they all tend, when you put them on, some of them are going to perform better over here, but this is the total of plants. All plants in total. This is the total of nutrients, all nutrients in total. Okay? Now, we will, in fact, be talking about specific plants versus specific nutrients. And let's talk about the nutrients first. This is a chart showing the availability of specific nutrients. Okay? As I move over here, you can see acidity. This is proton concentration, very large. As these arrows get, or these bars get wider, I'm seeing more availability. Okay? So acidity versus alkalinity. As this bar gets wider, I'm seeing more and more protons. Hydroxide, sorry. Okay? Now, the width of the bar basically represents the availability of that individual element or chemical, whatever, hydroxide. Okay? So you'll note that generally in the middle, pretty much all of them are somewhat narrow. But when you add all of the middles up together, that's why you get that large peak here. Okay? It's all of them in total. Another way of looking at this is this. This is out of your book, but these are those bars again. And you'll note that, yes, zinc is much more abundant here and a lot less abundant there, but it's got that much there. And the barium, boron actually tends to be higher in the middle. Copper tends to be higher in the middle. Molybdenum tends to be higher over here. Iron and manganese, much higher at this end. Aluminum, much higher at this end. Aluminum also higher availability here. Phosphorus, bracketing, calcium, magnesium, getting bigger at this end. You're adding all of them together, and that's where you get this curve. That's where you get this curve. Does that make sense to everybody? All right. So, if you look at all these nutrients, it turns out that at the high pHs, macronutrients, the availability, at high pHs, the availability of macronutrients is maximized. Okay, so when you're at a low pH, the availability of macronutrients are limited. Okay? At high pHs, the availability of micronutrients are limited, and at low pHs, the availability of micronutrients are high. Does that make sense? A different way of saying it. Low pHs, lots of micronutrients. High pHs, lots of macronutrients. Does that make sense? Okay. Now, how does this in fact affect plant growth? Well, there can be some direct impacts. Foliar damage when I have really high acidity, sort of if I have acid rain or something like that, the acid is actually literally going to damage the plants. And I will also have root damage at pHs that are lower than 4. Indirectly, and this is actually more importantly because our pHs generally, yes, we do tend to have pHs at these ends, but generally most of our soils tend to be in this middle zone. Okay? So we're really not going to have a lot of direct impact of acidity on our plants, but we're going to get a lot of indirect acidity. And most of that has to do with either too much of something or not enough of something. First example here we have aluminum, where the aluminum, I have too much aluminum, and the aluminum is going to cause stunting in my plants. If I don't have enough of something, and the example here is calcium, I am going to have stunting in my shoots and tops. Manganese can also be toxic to plants, and I also can have issues with molybdenum deficiencies. Okay? I'll show you some examples of this. Now, in general, plants try to adapt. Okay? And certain plants are more adapted at certain parts of the environment than others. Legumes are generally intolerant of acidity. On the other hand, maize is fairly tolerant. So different plants will move into different environments and be more successful or less successful based upon these issues, less this, but mostly this. And more importantly, this, the availability of those nutrients. Plants have a considerable range that they can live in. And this table right here basically shows you a variety of different plants and their optimum ranges of pHs. This is strongly acidic. This is slightly alkaline, slightly sort of mid-range, 6 to 7 plus. Okay? These plants down here at the low pHs, these tend to be plants that have either adapted or require something that's available at the low pHs. Does that make sense? Things like blueberries, pine trees. Species with high calcium needs require high pHs. Why? Calcium's available at high pHs. Okay? Species that have optimum ranges in the lower pHs, they're typically forest species from humid regions, and they have either developed, probably both of them, they have developed a tolerance for high aluminum, because there's low pHs, there's going to be a lot more aluminum in the soil, and they also probably have a requirement for higher amounts of iron. This is the evergreen trees. Okay? It turns out that most of our cultivated crops are adapted to the slightly acidic range, 5 to 7. Okay? Why? Happens to be where we have selected those crops from. The environment that those crops come from tends to have a pH of 5 to 7. That's not all of them, but many of them. Question in the back? No? All right. So let's take a look at some of these toxicities. This slide is terrible because you can't see the bottom slides, but this is pH down here, and this is calcium. Okay? This white space right in here is basically sort of the optimum availability of calcium. It's at the range of basically neutral pH. But what you're seeing here is this black line, but all these spots basically represent calcium availability as the pH goes up. All right? Now, let's look at the response in plants. This is from your book. This is a plant that has calcium deficiency. Look what's happening to its root structure. Okay, now this is not a toxic effect. This is a lack of calcium. Okay, so positive, this is the presence of calcium, no calcium. Another slide from your book. Basically, this is a calcium or boron deficiency, but you can see the twist in the corn. Direct response to pH and pH's effect on the nutrients, the nutrient availability. Okay, now that's the sort of the calcium end of the spectrum. Let's actually take a look at the iron end of the spectrum, or the lower end. Okay, this is out of your book as well, and basically what we're looking at is responses to aluminum. Okay, now this slide right over here basically shows a positive response to calcium hydroxide or an aluminum sensitivity, and these are negative or no response. Okay, and this is basically the root response. Basically, a higher number basically means that I'm getting a better performance. Okay, here are the plant species: honey locust, white spruce, gray dogwood, cedars, hickories, sugar maples. Okay, I start with the soil. Does it say it's pH? No, it starts at an initial of 3.8, okay? And I start adding calcium hydroxide, a liming agent, and seeing what's happening, okay? You can see a huge positive response as I start liming these soils, which basically says that these plants are having serious issues due to that pH, primarily from aluminum sensitivity. Now, these plants over here, when I start liming, I get a negative response. Now, is that saying that they like aluminum, or is it saying something else? What happens to my micronutrients when I start raising the pH? What am I going to be doing to things like iron? It's going to be less available, and as it becomes less available, it's not necessarily the issue of the aluminum. They have the tolerance. But because iron becomes less available, my plant's performance decreases. Andrew?
When you add the calcium, why is aluminum less of a problem? Is it just being attached to the soil solids and it's not available anymore?
Well, if I start raising the pHs, where's the aluminum going to be going? Probably the cation exchange, or potentially being buffered out or lost from the system. Cool? Good example of this response. Micronutrient need, macronutrient need. Here's a response of, this is actually what aluminum does. Okay, what it does is it basically blocks calcium transport into the plants. Okay, and it binds with this aluminum will bind with the ATP that's in your body. I mean, ATP is the energy of the cells, right? Okay, if aluminum binds with it, the plants can't use it, and they can't get the calcium into the system either. So as a result, what I have, this part of, don't look at this part of it. As a result, what I have is I have problems with plant basically growing. The cells won't expand, I can't get calcium into the system, and I don't have any energy. Okay? Now, this chart basically here is the aluminum in the soil solution versus the soil, it's just the soil pH. Okay? Just so that you can see what's going on. As the pH goes down, the aluminum concentration in the soil solution dramatically increases. Okay? Now they also threw this one in here just so you can see sort of this blip of this reaction. Okay? pH here is about 4 or 5. Right about here is pH 4 or 5. As the pH drops more, we lose this, it moves to this. As the pH drops more, we lose this, we move to that. So basically what you're seeing is you're seeing that response of aluminum going into solution through this sequence. Okay? Questions? Cool? Now, interestingly enough, if you look at this, it turns out that aluminum is rarely a problem when pHs are above 5.2. Well, why? Above 5.2, there isn't any really aluminum in the soil solutions. All right, let's look at aluminum. This is what happens to the plants. These are tolerant plants versus sensitive plants, tolerant versus sensitive, and you're basically looking at different pHs. Okay, this is pH 4.4. This is pH 5.2. Remember, at 5.3, 5.2 is where we start seeing those responses. Sensitive plant at 5.7 looks a little bit less than the tolerant plant, but not that much. You get to 4.4, same species. Look what happens to the plants. I can't pump in calcium. The aluminum's blocking that. The aluminum's interacting with my ATP, so I can't use the energy. And then finally, it resists or prevents me from enlarging my cells. And this is the type of plant that ends up with, if you have all three of those things hitting you. Questions? Cool? Now, from a management sense, okay, we're looking at aluminum still, okay, and this is soil pH, but we're looking at here is two different soils, one with no organic matter and one with organic matter. Now, we're trying to start integrating some information here, okay. Why do you think I'm having, with no organic matter, starting at pH 6 down to about a pH 4, look at the amount of aluminum that are going right into solution versus the 10%. What's going on? What? Aluminum is binding to the organic matter. Why in specific? What happens when the pH goes down and you have a pH-dependent charged molecule? Remember this? Okay, let's imagine this is organic matter. It's attached to an O. Here's the end of the structure. Okay, this is our carboxylic or something like that. What's going to happen when the pHs start to drop? pH is going down. My concentration of protons are going up, right? What's going to happen as the pH goes down? What's going to happen to my pH-dependent charge? Any thoughts? You guys remember pH-dependent charge? It gets more negative, right? Does it? You guys tell me. As the pH goes down, what happens to pH-dependent charge? What happens when the pH-dependent charge? What happens when the pH starts going up? If the pH starts going up, I'm going to be increasing the concentration of OH, right? What's that OH going to do? No bites?
The pH goes down, the charges are more positive.
As the pH goes down, the charge becomes more positive. The proton's going to add here, right? Correct? No? Yes? No? So what's going on with the aluminum? I should not have gone this way, should I have? Is it just too early in the morning? What? No? Well, the aluminum's, what's going on? The answer is correct. The answer is the aluminum is going to be binding with the hydroxide. Right? Okay, the aluminum is going to be taken out of solution. So as a result, as that pH starts dropping, forget about that. Okay, as the pH starts dropping, I get more and more aluminum into soil solution. Okay, that aluminum with no oxygen, with no organic matter, is going to go straight into toxic effects. It's going to start interacting with these plant roots. It's going to stop pumping calcium or preventing calcium being pumped. It's going to bind with the ATP. It's going to prevent cell elongation and cell enlargement. Okay? But I have organic matter. What's going to happen? That aluminum is going to bind with the aluminum is going to bind with the organic matter, and it's going to reduce the amount of aluminum that's on the soil solution. And so as a response, I'm going to have less of a toxic effect. Does that make sense? Yeah? Okay. So the organic matter is, in essence, it's not truly a buffer, but in essence, it's acting. It's not a buffer for the protons. It's a buffer for the aluminum. It's binding with the aluminum.
So why is
the pH isn't lower with the organic matter. At this pH, pH of 5, when I have organic matter, when I don't have organic matter, we're basically seeing that I have different exchangeable aluminum. Okay? Read it this way in that sense. Go.
And that's because the pH dependent charge of the
Yeah, I think I shouldn't have gone that direction. I think, yeah, I want you to think it's really binding. The aluminum's binding with the organic matter. Okay, we feel good about this? Kind of-ish? Okay, questions? No? All right. Other toxicities. What other things can happen? Manganese toxicity. It's a nutrient, but it can become toxic at high levels, 5.6. Basically, we're looking at tissue manganese, and this is different pHs. And you're basically looking at yield. This black line is yield. The green line is the amount that's in the tissue. And as the pHs start dropping, we start seeing more manganese in the tissue. Our yields are dropping, which basically means it's a negative plant response. The plants are not having as good of time. Okay, does that make sense? Okay, there are other toxicities with protons themselves. Forget about the direct damage. You can have that, but it also potentially will kill microbial populations that are beneficial to the plants. Okay, as you lower pHs, you're going to be changing your microbial communities. All right, last slide. Questions? We're good? All right. Go, question.
So the aluminum and manganese, are those common for field crops, or why is it that
Aluminum, magnesium, are those the two most common?
toxicity?
Toxicity. Most toxicity issues, the biggest problems with nutrients is really availability. It's less toxicity events. Okay, so your first responses in plants are not going to be toxic responses. The first responses you're going to get from nutrients or from pH has more to do with availability of the nutrient. Is there a nutrient that they need that's not there? Okay, when you start getting to the extremes, that's when you start seeing the aluminum and manganese and calcium issues. Okay, toxicity is not necessarily, generally we associate toxicity with a direct impact, but use calcium as an example, where if I don't have the calcium, I mean, you saw the slides. If I don't have the calcium, I'm basically going to get the same response as I would with aluminum. I mean, look at these two slides. There's the aluminum slide. Okay, look at this part of it, okay, versus that. Okay, one's an aluminum. This one happens to be the lack of calcium. One's an aluminum. I'm getting, in essence, it's not the same response, but the morphologic feature is basically the same. Okay? So, I mean, they're both, I'm getting stunted roots. Okay? Now, the cause, in this case, is because I don't have something, and in the other case, it's because I have too much of something. Okay? So this is, the second one, the aluminum, that's considered a toxicity event. This is considered a deficit event. But the responses are very similar. Was that, am I going in the wrong direction?
No, that makes sense. So calcium, aluminum, and manganese are the most.
Oh, you want to know which are the most prevalent?
Yeah, I mean, why is it that you chose those three?
I chose those three because they're very easy to see. Okay, there are a lot of toxic events. There's a lot of toxicity issues, and then there's a lot of deficiency issues. Okay.
And I have kind of a follow-up question. So if it's deficient in calcium, would that indicate that it's potentially toxic, have a overload?
So potentially, yeah. So the question was, if it's deficient in calcium, does it mean that it's got toxicity in aluminum? Okay. As the pHs go down, we can go back to this slide, like this one. No, let's use this one. Okay. As the pHs go down, that's when I'm going to start losing the calcium. But as the pHs go down, that's also when I'm going to, I don't see aluminum in there, but oh, there it is. That's also when I'm going to start seeing aluminum. So there's going to be some bridge point where I'm seeing, some point at which I'm seeing both the deficiency of calcium and the toxicity of aluminum potentially in the system. Is that what you're asking? Okay, go.
Why is there aluminum over there?
Turns out aluminum availability is high at both ends. There's actually a lot of them that have high availability at both the high and the low pHs. Okay, there was another question out there. Hopefully got answered. No? Okay. We feel good with this so far? Because I'd like to go to, before I let you guys go, I want to talk about this one slide. Okay. What we've been talking about for the last 3 weeks, and we're going to be talking probably about for the next 3 weeks, is in essence this slide. We're talking, this is an environmental slide. We'll walk through this slide. But conceptually, what I want you to look at here is how these things are interlinked with each other, how these systems are interlinked. Okay? Now, we've been mostly talking about what's going on here down to here. Okay? This is the soil solution, the pH, okay, the water. Okay? The exchangeable reactions with that water with the colloids and potentially weathering reactions. Okay? So this would be, in essence, the sort of the reserve, not the reserve, the residual, so the structural pH. These right here would be the exchangeable pH, and this would be the soil solution, the active pH. Okay? So that's what we've been talking mostly about. But we have to think about where this stuff comes from and how it interacts with the larger world. Okay? The model system we've been talking about is plants. Okay? What's the response of plants to this? Okay? And what's driving this through its exchange complexes and through weathering? Okay? But what are the feedback mechanisms this way? And how is management playing a role in this soil solution? Okay? It's a picture of a tree there. But that easily could be a picture of a corn plant. It easily could be a coffee tree. It could be easily any kind of plant you want. Okay, so let's walk through this. We're going to start here and we're going to end here. Here's the soil solution. This is controlling the pH. Okay? This pH is controlling the nutrient availability. Okay? Those nutrients are then taken up by plants. Okay? Now we can harvest this and move it off a system. We can feed it to animals and bring that stuff back into the system. But we're certainly going to get a certain amount of soil organic matter from the system. Okay? That soil organic matter is going to be put into biomass. That biomass can be recycled into new biomass, microbial decomposition processes, for example. But it also can be mineralized. This mineral material can go into the soil solution, which then can be taken up by plants, or it can be immobilized and processed here. Okay? As these things move this direction, they're going to be controlling the pH and the nutrients that are exchanging on the base saturation or the cation exchange sites. Okay? Now, also when we're harvesting in an agronomic system, we're certainly going to be imagining that we're going to be depleting the bases that are available here. If that's a system, we need to be able to bring in material. Okay? In this system, we're looking at particulate matter and liming, as well as organic matter. This could be easily fertilizer. Coming in as solid material where it weathers and goes into soil solution, back up to our plants. Okay? Does that make sense to everybody? All right, we're going to be expanding on this in a little bit for the next 3 weeks. Be free, guys.